Hello. This lecture is on PCR components. It's part of the DNA amplification lecture. It's the second half. We've already gotten halfway through it before spring break in the semester 2020. So this is going to be a strange video for some people in this course. Anyhow, I'm Jason Linville, and this is being recorded for CJ352 Forensic Science Lab 2, which may soon experience a name change as somebody recently brought up because there is no Forensic Science Lab 1. So anyhow, let's go ahead and get started. This is what we're trying to accomplish today, and as I just gave a long sort of diatribe to everybody here that I'm not going to repeat, these are new learning objective slides that you guys should use to sort of predict what to study for when you're preparing for quizzes and exams. All right, so we're going to go, we already went over a couple of these slides right before spring break in this class. So we went over sort of what are the 4 PCR components, and then I mentioned with this slide, there's also a couple extra things in there to get the ionic and the pH right, but who cares about that? We had a long conversation about Dom DeLuise and whether he was or was not in a Prego spaghetti sauce commercial, which has nothing to do with this course and won't be on the quiz and exam, because you'll notice Dom DeLuise was not mentioned on that learning objective slide. Okay, our reaction is going to take place in a tube. So if we were actually to do this lab in class this semester, which unfortunately we're not going to be able to do, we would get out the 4 different reagents. Some of them would be mixed into the same tube, and we would just squirt all of the things into this tiny tube here. Let me make sure my little pen is on. Get out of there. Is that it? Yeah. Put it in this tiny tube, but when you prepare something for amplification, you need to make sure you're in a very clean area, because a little bit of DNA, either from the analyst or from the environment, can then be amplified and interfere with your results. So we have this little sort of PCR hood here that you can keep very clean on the inside, and when you turn it on, it creates a little wall of air to separate the inside from the outside, and has a UV light that you can use to sterilize everything after you're done. The one thing important here is this PCR reaction is normally set up in an area separate from after the DNA is amplified. Because remember, we're not amplifying all of the DNA in the tube, just specific portions of it. For forensic science, it's those short tandem repeat areas. And once those areas are amplified, you have 10 million copies. If that amplified DNA gets back into the prep room, then only a very tiny amount of that amplified DNA can cause interference. So you have to be very careful to separate your preparation area from your samples after they're amplified. And usually that's done physically in a different room. Okay, so what we're going to do is we're going to go through each of these components, and I'm going to give you a little bit extra information about them. So not only do you need to describe them as far as what DNA does in a PCR reaction, but also there's a little bit extra information about each of these things, including why is he still here? Okay. All right, number 1. DNTPs, okay, these are the letters. So to clarify, when we say DNTPs, okay, that is the same as nucleotides. That's the same as the AGCs Ts. Okay, now what the DNTPs actually is, if I can interpret this for you, that N is a placeholder where it means it's a mixture of A, G, C, and T. It's basically any nucleotide. So DNTPs, the D is for deoxy, because you could also have ribo NTP nucleotides. Nucleotide triphosphates is the thing there. So it's a mix of DATP. It's a mix of DGTP. But what I want you guys to think of these is just think of these as the loose letters, the A's, G's, C's, and T's. Okay, we're not going to go over the reaction of how 2 phosphates are lost and that supplies the energy for the addition of the letters. No, I just want you to see DNTPs and know it's a mix of A, G, C's, and T's. Okay? Okay. All right. The Taq polymerase. Now, in the description of what goes in, the 4 things that go into the tube, you get credit if you say DNA polymerase, you get credit if you say Taq polymerase. But in these next 2 slides, I really want to clarify what the difference between DNA polymerase and Taq polymerases. Taq polymerase is a special type of DNA polymerase, okay? So in a polymerase chain reaction, okay, when it was first demonstrated in 1985 by Kary Mullis, won a Nobel Prize for it, what his team did was they actually used human DNA polymerase in this polymerase chain reaction. So what do we know about PCR? We heat it up, the DNA comes apart. Okay, this is a little bit review from spring break that everybody's forgotten. Heat it up, 94 DNA denatures or comes apart. Cool it down to around 60 degrees, the primers attach. Then you heat it up to 72, the polymerase attaches and adds the DNTPs. We've covered the 4 different things in a PCR reaction. So you could do that in a laboratory. DNA comes apart, okay, anneals attach, elongated. But when using human polymerase, in that next part of the cycle where you would heat up the reaction to 94 degrees again, the DNA polymerase would fall apart because human DNA polymerase denatures or it falls apart at high temperatures. But they could still run the reaction because in 1985, what they did is they had a bunch of graduate and undergraduate students. So they would heat up the reaction, they would cool it down, allow the primers to anneal, heat it up to 72, open up all of the tubes and add DNA polymerase, close them and allow the elongation to progress for 2 minutes, okay, or so. And then they would heat them up again. The DNA polymerase would fall apart, the DNA would split, cool it down, the primers attach, heat them up to 72, and have that student open up all the tubes, squirt, 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 all of the polymerase. It was a very they would have to sit there for like 3 hours, and 28 times they would have to open up the tubes and squirt in the polymerase. So it was a very time-consuming process when PCR was first demonstrated. It was expensive because polymerase does not grow on trees. Actually, it grows in our bodies, but anyhow, that's neither here nor there. It's expensive to extract, right, and sell. So what scientists did when they realized this, that human polymerase would fall apart and they would have to be added at each elongation step, which was bad because it was time-consuming and expensive, they thought, okay, maybe we can find, because DNA has to be replicated in all types of living things, right? Whenever a cell divides, a DNA polymerase is there. So maybe we can find some other organism that survives at high temperatures that might have a polymerase that when we heat it up to 94, it does not fall apart. We can just put it in the tube at the beginning of the reaction and let it go. And scientists discovered that Taq polymerase was a polymerase that met this description. Notice the Taq there, T for thermos, where's my little thing here? T, AQ for aquaticus. Okay, this is a bacteria that survives in hot springs. It survives at extremely high temperatures, and the polymerase for Thermus aquaticus did not denature at 94 degrees. So you can just add this DNA polymerase, which we now call Taq polymerase. So in my questions and on the exam, again, what are the 4 things in a tube? You can give me either answer. But if I'm asking you about the enzyme that does not denature at high temperatures, human DNA polymerase is not the right answer there. It's Taq polymerase, okay? So you order Taq polymerase, squirt it into the tube as one of your 4 components, and you just let that PCR cycle progress. You don't have to add new Taq in each cycle. Okay, here we have some primer, and I'm thinking I changed some of these slides around. If you printed out the PDFs a little bit earlier, you may find some of these slides are a little bit different. I tried to take out the non-important information and highlight the important information. And then the primer slide is one that I changed. Okay, primers are short strands of DNA, so description of primers, short strands of DNA that will attach to each end of the location to be amplified. So it's important to understand that PCR, you run the reaction pretty much the same when you want to amplify DNA. You need to put in DNA and primers and Taq polymerase and DNTPs. But when you amplify DNA, whenever you run a reaction, you usually want to amplify different areas of DNA. If you're studying genetic diseases, you want to amplify the gene in the DNA of the disease that you're studying. If you're trying to do species identification, you want to amplify a protein in the genome that's different among species. If you want to amplify an STR, you need to locate that area in the genome with those repeated base pairs and amplify that area. So you need to choose a primer that will attach to the DNA on the upstream side of the gene you want to, or excuse me, the area of DNA you want to amplify, and another primer that will attach on the opposite end at the other side. So that way, when your Taq polymerase sits down at the ends of the primers, it's going in the correct direction. Okay. So the sequence of the primers affects the location of the DNA that's amplified. So in other words, if I want to amplify D5S818 or whatever, an STR, a short tandem repeat location that will give me one pair of numbers, okay, in the human genome, I look up the human genome in that area and I order a primer that will attach to one end it, attach to another end it. I tell a company who makes primers, okay, they manufacture it in the lab and then they send me a tube with, millions of copies of that primer in it. And then that is what I squirt into my amplification tube, one of the four components when I add the primers. Okay. All right, and this sort of shows you the DNA splitting apart. And we're really not going to go into a lot of details about 5' 3' here. But originally, this G is attached to the C, G is attached to the C. But you didn't heat up the DNA, the two strands split, the ladder breaks, right? And then the primer attaches to one of those strands at one end, okay? And then this DNA is sort of connected down here, and this DNA is connected down here, okay? And then the other primer attaches to the other strand at the other end, okay? And then the polymerase is going to sit down right here. Why are these lines going all squiggly? Yeah, hold on, I'm trying to do something creative here. Erase all link on slide. Okay. And then when your Taq polymerase sits down, the Taq polymerase is going to sit down here and go in this direction. And on this primer, it's going to sit down here and go in this direction. Okay. All right. So when you are designing these primers or ordering these primers, it's important to know that the other thing I want you to understand about these primers is when we describe the three temperatures of PCR reaction, we talk about 94, the DNA splits apart, the annealing temperature we set around 60 degrees, and then elongation at 72. I do want you to understand that that cycle and those exact temperatures, specifically the annealing temperature, which is where the primers attach, the annealing temperature for primers is different depending on the sequence of a primer. So in other words, if I'm running a reaction where I'm amplifying an STR location, I may want to set my annealing temperature at 62 degrees, okay? But if I'm running a different amplification reaction where I'm amplifying the cytochrome oxidase gene in flies for some reason, I may want to set that because I'm using a different primer set at 55 degrees, okay? And what I took out of this lecture is there are certain problems that occur if you set that annealing temperature too high and other problems that occur if you set that annealing temperature too low. But what I want you guys to understand is recognize the annealing, that second temperature in the second temperature in the cycle, that's where the primers attach, and that needs to be adjusted for every reaction you do whenever you switch the primers. Okay, the primers will come sort of with its annealing temperature or suggested melting temperature on it, and you set the annealing temperature based on that. Okay. All right, so, and there's a great picture of it. So in this picture, notice I keep saying 60. In this picture, it's probably around like 53 or something like that. This annealing temperature can change, and be anywhere in this range, and where the annealing temperature actually is based on the sequence of those primers. Okay, thermal cyclers. So this is the instrument after you put all of your components together, which we'll go through an STR demonstration. Again, I'm still trying to figure out what this lab is going to look like, but we'll sort of see what the reagents look like here. After you set up your reaction, put your DNA, it never hurts to say this too much, put your DNA primers, polymerase, and DNTPs into the tube. Then you put that tube into this thermal cycler. And this thermal cycler is just an instrument that contains a metal heat block that the plastic tubes settle into. And when that metal heat block, the temperature goes up, the temperature of the reagents, the liquid in the tube goes up. When it goes down, they go down. So you have this heat block hooked up to a small computer, and actually not a desktop computer, but the computer is just like in the instrument itself. And you just program into the instrument, as far as I want the heat block to go up to 94, stay there for 2 minutes. Go down to 60, stay there for 30 seconds. Go up to 72, stay there for 60 seconds, okay? And then just do that 28 times. And then after you're done, go down to 4 degrees and hold it at refrigeration temperature. Okay, so this just sort of gives you the name, sort of describes what that thermal cycler does, but it's basically to change the temperature. That's how so it's really not that fancy. This thing about the heated lid, this probably isn't really important, but I do want to mention, if you just threw a tube without a heated lid into the thermal cycler, what would happen is you would heat up the reaction to 94, and when you cooled it down, you would get a lot of condensation occurring, just water occur at the top of the tube, the cooler area of the tube, if it was exposed to air. And that sort of, it's like evaporating all of the water out of your reaction. Now all of your components are too concentrated in what's remaining at the bottom of the tube. So that lid is heated, it just sort of keeps that condensation from forming, and it keeps everything sort of, some of your liquid will go into vapor form, but it keeps everything at the bottom of the tube. Helps with the efficiency of the reaction. This sort of shows you an example of what some thermal cyclers look like. This is an old one, but if you walk into a lab, unlike some of the other pictures I have, there's a good chance you're going to see a thermal cycler that looks like this, because these things, they tend to be workhorses. They don't break down. They work for a long time. So it's not unusual to find a 20-year-old thermal cycler still working in the lab. You can see here, when I say computer, it's like the old-fashioned push button type menu selection button. But this knob, you unscrew, and this whole tray slides back. And then you put your individual samples, which sometimes in larger labs that run high throughput, a lot of samples at one time, there'll be this tray where you can see these are just a whole bunch of tiny little tubes there. And this whole tray goes in the instrument right there. And then whoop, the lid slides back, and you hit the start button, and then you can set it and forget it. That's right. I have no idea why I suddenly turned into Ron Pompeo or whatever his name is. Okay, then you walk away, and then you come back when your reaction's done. Okay, so the tubes are, I don't know, for you numbers, the tubes are small. I'd show it to you if we were physically in class, but they're a little bit, or they're smaller than the tubes that we use for our extraction. And then the reactions are usually a small part of that. Okay. All right. The last thing I want to talk to, so notice there we have another transition here into this, what am I talking about? Into a learning objective slide. Okay, so we're going to talk about what a positive and a negative control is. And I want you to explain to me because a positive control is where you put DNA interaction, it should be positive. I want you to explain what information you can get when a positive control fails. In other words, it's negative. I want you to be able to tell me what it indicates when a negative control that should not amplify because it doesn't have any DNA in there, what it indicates when a negative is positive. And this is one of those things, just to forewarn you, that I'll say in a question on a quiz, you run amplification reaction and your negative control is positive. What does that mean? And the student's sitting there going, well, it either means something's not working or there's contamination, and you just take a guess. Okay, actually take the time to sort of visualize what's happening in the positive and negative controls, and then you'll better understand and better be able to explain what it means when your positive is negative or your negative is positive. Or I may ask you, what if your positive is positive? Well, that's what's supposed to happen. So what does that tell us? Okay, let's start with the positive control. All right. Now, if we run an amplification reaction, put it on the instrument, and just there's no DNA there, like we did not amplify DNA. There's a lot of explanations to that, right? Especially if we're working with evidence. It might have been where, well, we screwed up our extraction, so there was never any DNA in our amplification reaction. It might have been, oh, the evidence that we collected, there wasn't any DNA in that evidence, okay? It was a very small amount of DNA in the evidence. So that's why there was no DNA in the extraction and no DNA in the amplification, and it didn't work, okay? The reason for an amplification positive is because we don't want to have a mystery surrounding, oh, was it the amplification or the extraction or the evidence that did not contain DNA? We want to run a sample where we know we take DNA out of the freezer. It's not an evidence extraction that should have DNA in it. It's not even a known sample that we extracted from a person that should have DNA in it. It's a tube that says DNA control on it. We buy it from the company. They were certified. The sample is certified, and we are confident that there's DNA in that tube. So when we put DNA into the reaction, we know every single time we run that positive control, we should get DNA amplified, and it should be a specific profile that matches the cell line of the DNA we bought. Okay, so an amplification positive control, which is a little bit different from an extraction positive control. An amplification positive control, it's an amplification where we know we've added DNA in there. Okay, so if we have successful amplification and we run this reaction, then that just tells us everything's working. Okay, it tells us the reagents are good, the DNA is good, and the reaction was performed correctly. Okay, Ariel asks if the bot DNA can expire. It has an expiration date on it, and when you're running a accredited laboratory, you want to abide by that expiration date. But with that being said, DNA is such a stable molecule. If you keep the positive control in your freezer, especially if it's one bought by a company that you know is very clean, it doesn't have any nucleases in it, it will last for a very long time, like 50 years type of a long time. So in general, even though it has an expiration date on it, you can use your positive control for a long time, especially if you're in a research lab. Ooh, that's a good question. So Daphne, where these companies, so Daphne asks where the company gets the DNA from. These companies typically, they will grow human, especially, and let's just stick to forensic science, like human DNA, known human DNA. They will have cell lines that will grow, that they can actually grow in culture. Okay, so oftentimes these are mutated human cell lines, because you can't just take like cells from your cheek and then grow them on a petri dish and they live forever. So these are cell lines that are they're able to grow in the laboratory, that they can keep going. So there's a couple, so they tend to have funny names like of cell lines. So there's like K562 is a cell line, 9947A is a cell line, 9948 is a cell line. So there's these different cell lines that they have, and then they actually just extract the DNA from those cell lines. So when you buy 9947A today, you get a profile of a certain 13 pairs of numbers. And when you buy that cell line 3 years from now, it's the same profile with the same 13 pairs of numbers. Okay. And it's a good question, because sometimes you think, oh, well, they probably just take a person and extract their DNA and then they sell that. And there are some known standards that you can get that I think are from maybe different donors or combinations of stuff. But most of the positive controls that come with kits and forensic science labs use, it's a cell line that always has the same profile. Okay. So if everything works, then we know that, and there's going to be a theme here, the reagents are working, the instrument is working, and the person has done it correctly. If your positive control fails, then that means either there's a problem with the reagent, a problem with the instrument, or a problem with the goofball who was setting it up. Okay. And when I'm running reactions in our laboratory, students who are doing this they always assume it's them. They always go, oh, my reaction, there's something wrong with me. No, oftentimes it's like we use the same program. It was an instrument problem. Okay, we used the wrong reagent. The reagent has gone bad, or something. It's a reagent problem. Okay, so it can be those three. All right, what happens if you get a failed amplification? That's something that is not supposed to happen. If somebody could answer my question in the chat box here, what should you do if something goes wrong? Anybody? This is a life lesson, remember? Run it again. Very good. Okay, so you do it again, and then that oftentimes will give you you do it with a different person, you do it on a different instrument, you do it with different reagents, and then oftentimes that gives you, and if it happens again, okay, then you complain. Normally you have somebody else run it, maybe on a different instrument, or if this instrument has been working for all other samples, but just not for this one, usually you can narrow it down to a potential problem with the reagents and you can complain, okay? But again, if a positive, this is the quiz question, if a positive control fails, what does it indicate? It indicates that there is either a problem with the reagents, the instrument, or the scientist, okay? Something went wrong with the reaction. All right, a negative control is an amplification reaction you run and you don't put any DNA in it, okay? You just squirt water in there that is nuclease-free, doesn't have any DNA in there. So what's supposed to happen is nothing, okay? Negative control, you put it in the thermocycler, run it through, you load it on your instrument, and you get no amplified DNA, no human profile. That's what's supposed to happen. Okay, so a negative result or a negative control that's negative, it indicates that the reagents were not contaminated. There was no DNA in the TAC, no DNA in the primers, no DNA in the pipette that you were using. Okay, again, the reagents were not contaminated. The pipette, the equipment was not contaminated, and the person was not, sneezing into the thing. So the person didn't contaminate it, okay? If you get amplification of DNA in your negative control, it means it either got in there in the reagents, the equipment, in this case, the pipette, or the person, the scientist, okay, by sneezing or coughing. Or another thing down there, human error, okay? Instead of adding the water, they accidentally added the DNA, or they accidentally mixed some DNA in with one of the reagents. So it could be human error as well. Okay, so quiz question, what happens if your negative control is positive? That indicates contamination. So don't tell me if your positive is negative, that's contamination. Or if your negative is positive, that something went wrong. I want you to be specific when a negative control turns positive and tell me it's contamination. DNA is in there that should not be. All right, finally, how do you prevent this contamination? How do you keep your negative controls negative? Well, as it says here, it could come from the scientists, the police, other people setting up samples. We went over, I think, the difference between mixture and contamination. All mixtures are not necessarily contamination. This is DNA that gets in there after or during the sample collection. And a way to prevent that, like I mentioned before when I was describing the setup areas, is you keep your amplification area separate from your setup area. So the pre-PCR, that means your setup area. Before it goes into the thermocycler, then you physically take your tubes into another room and put them into the thermocycler, and that is your post-PCR region. And then in the same room as that thermocycler is also all your equipment to do the analysis after amplification, okay? There's all the other steps, irradiate your PCR setup space, clean your workspace with isopropanol or other things. I'm not going to ask you for the specifics of that. The other thing down here, a good way to prevent contamination, and this is an important one, is you never want to have, well, actually, no, we weren't working with that, so I didn't mention this during extraction, but this is true for extraction as well. You never want to be working with your known sample from your suspect at the same time as your evidence sample, because if you accidentally get the known person's DNA into your evidence, that's the worst mistake a forensic scientist can make. Then it looks like that person was at the crime scene, implies they were guilty of a crime that they did not commit. Okay, so in order to protect that suspect, you keep their DNA, their cheek cells, in the freezer until the evidence is done. You always process your evidence, your unknown sample. So therefore, if I'm suspected of a crime, my DNA is in the freezer, they haven't touched it, okay? They process the evidence, get a profile that matches me. They know it's not a mistake where they accidentally got my DNA into the evidence, okay? It wasn't open at the same time. It wasn't even open in the lab. until they got that profile from the evidence. Now they can take my DNA out, process it, and see if it matches. Okay. All right. That's it. Okay. What I'm going to do now is I'm going to keep the recording going a little bit. You guys can ask me questions in the chat box. At this point, if anybody wants to turn on their microphone and ask me a question, you also have my permission to ask me a question with your human voice. Okay. And then if there are no questions, or after a couple questions, then what I'm going to do is stop the recording, and then you can ask additional questions that will not be recorded if you prefer that. So I'll say I'm glad to see so many people attended today. The other thing is, if you want to leave right now, now's the time to leave, because it's the important part of the lecture is over. Although sometimes the questions are the most important part of all. I think I mentioned in that welcome back or transition to online video, I mentioned that I'll probably record a separate video about grading policies. I was speaking to my class earlier today, and I think you guys got an email about the potential for choosing pass-fail options in some of your courses this semester. I know there's currently still a lot of debate, so I'm not sure how much information has been provided to students, or if they're still tweaking exactly how this is going to work. But the one thing when you do get that email so Ariel asked the question, for this course, I am going to allow that option. And I just want to remind everybody, the reason that option is there is because I'm sure everybody in this room has experienced, sometimes if you're fortunate, then you are in a location where you have food and shelter, and you just have to stay put, internet access, and you have to stay put. For other individuals, your entire schedule can be, completely turned upside down. So it's really for those individuals that just want to, sort of take a retake and choose, okay, I'm just going to do pass fail on my courses. You have that option. For grades, and I'll answer the Marlo's question here in a second. For grades, what I don't want to happen is the day before the last day of class to have a bunch of people saying, am I going to get a B or a C? What do I need to get on the exam? And I'm like doing all these calculated grade calculations to help you choose whether you want to pass or fail or B or C or whatever. But again, for the situation, if you're really struggling this semester and you want to ask me, hey, Dr. Linville, what more do I need to do to pass the class? I'm really having trouble keeping up. You can send me that type of email, and I'll figure hey, do a couple more assignments, or just take the exam as long as you do halfway decent, on it, and review a little bit. If you're struggling, because like I mentioned, I don't know how I'm going to do these quizzes since I don't have a question bank for them. I may just email the PDF file to everybody. You fill it out and take a picture of your answers and then send it back to me, or submit it through Canvas probably. So there's still sort of, I'm just here to help you guys out for those that are in a challenging situation, helping you decide whether you want to go pass fail, and let you know what you need to do to pass the course. But from my perspective, the teacher's perspective, I treat at the end of the semester, I'm still going to submit an A, B, C, even for students that choose pass fail, A, B, C, D, or F. Okay. It's just that for students that choose pass fail, if I put in A, B, C, D, that gets changed to a P. Okay. So there's not a way to say, oh, well, if I get a B, I want a B, but if I get a C, I want a P. You're not able to do that. You just have to sort of, figure out where you're at before the last day of class and decide whether you want to pass fail. I should have your grades in soon. As I mentioned before, spring break was definitely not a break, and I spent a lot more time getting these classes and people and data and everything prepared. So I'm behind on all my grading. But again, you guys have a couple weeks to make that pass-fail decision. So if anybody if your first exam grade has to do with that decision, I will get those grades posted soon. No, you can't see I don't even think the thing is, if I unlock the door for the cats, then you'll end up seeing my children. And if you see my children, then something will end up being broken on the computer. Probably not. All right, hold on. All right, yeah, it's all right. So what I'm going to do, you guys asked good questions. I'm going to stop the recording, but I'm going to continue answering questions. All right, if I can get the menu up.